E ho mai ka ike mai luna mai e o nā mea huna no e au nā mele. E ho mai, e ho mai, e ho mai, e ho mai ka ike mai luna mai e. O nā mea huna no e au nā mele. E ho mai, e ho mai, e ho mai, e ho mai ka ike mai luna mai e. O nā mea huna no e au nā mele. E ho mai, e ho mai, e ho mai, e. Nā au makua mai ka la hiki a ka la kau, mai ka hoʻokuʻi a ka halawai. Nā au makua ia ka hina kua, ia ka hina alo, ia ka kau i ka lani. O ki ha i ka lani, o we i ka lani, nulu i ka lani, ka holo i ka lani. Eia nā pula pula a o ko o nā oiwi o Hawaii. E mālama o ko ia mākou. E ulu i ka lani, e ulu i ka honua, e ulu i ka pai aina o Hawaii. E ho mai ka ike, e ho mai ka ikaika, e ho mai ke akamai, e ho mai ka mau popopono, e ho mai ka ike pāpā lua, e ho mai ka mana.
Aloha, everyone. Welcome to the third session, Current Genomics. I'm here with Dr. Emmet Aluli, who will be moderating this session with our speakers, Dr. Leah Dowsett and Dr. Keolu Fox. We'll do introductions a little later. I want to start with just some of the housekeeping, just so everyone knows that this webinar is being recorded. And the recordings are available at Papa Ola Lōkahi's website, and that is listed on both the flyer and it will be posted during the evaluations. Please mute yourselves so that we can hear everyone clearly. You can have the question and answer box to post questions. And we will be having the staff at Papa Ola Lōkahi. Big shout out to the people behind the scenes who will be posting relevant articles throughout the talk. And we encourage you to complete the evaluation. I'll be sharing some of that today so that you hear what the feedback from sessions 1 and 2. So let me start with just saying that I appreciate those of you. I know there are some have been logging into all of the webinars, and I've heard lots of great feedback. We're so blessed to have our own Kanaka resident experts in this area, Dr. Leah Dowsett and Dr. Keolu Fox. In session 1, they covered Genomics 101, looking also at lessons learned and advancement in this field, bringing up the bioethical questions and circumstances arising in the field around policy, the technology, and data sovereignty. We're going to hear more about that today. We've been introduced to a lot of new terms. I know myself as a layperson, things like data sovereignty. We've talked about data in other research and community-based research, but it's interesting to learn more about how this extends to the work in this area as well. Indigenous-led genetic initiatives. Another term, biocolonialism, mining of our genomes, government research and big pharma, looking into use of our genomes for developing the next generation of drugs. I know we'll be hearing more about that today. Also learning about open access biobanks. What is that? Who has access and control of that? And the most encouraging is learning about what's happening with what Dr. Fox terms as paths forward. And I really appreciate that because it then guides us on how to support this effort, how to advocate for it, and how to drive it. So I wanted to just share some insights, and Emmet will share some as well, but just some 2 quick insights from me over the past sessions is just having learned colleagues to teach us more about current projects, how they're related to us, and how the history of this field of science has affected us and will affect us in the future. The other is just learning things about, genomic science. The big aha for me was this identified mutations and finding mutations in these large studies, and that how a lot of it has been affixed to us as ethnic groups, or when instead we should be looking at how they have been shaped. These mutations have been shaped by environment. And as Dr. Fox stated, we are the Aina in the land, and that environment has resulted in mutations that are common locally to that group, but then rare globally. But that, in fact, is the thing that is of great interest to large pharma companies. So the more we learn about it, I think the more informed we can be. So Emmet, do you want to share your insights? You have to unmute yourself, Emmet.
Got it. For me and for most of the Kauka who report, this is really an exciting and an important issue we're trying to get on top of. There was a comment at the last genomics webinar, and it comes from a guy named Manny Kulolo'io. And his concern was that Kaka'aku was designated for a biomedical sciences with tax credits and incentives. And with multiple companies and tidies that would have a substantial database for the so-called, he called it ground truth. It was also kind of looking at the U.S. Department of Defense priority mapping of the genome as a key determinator, like the prize, he's saying underwater signal processing of sounds and the detection of classification of passive solar object characterization. So his father and his family were the culture monitors of the Honokahua burials at the Ritz-Carlton Hotel. And it was the first time in history that they stopped for once all the free for all studies in their removal of the EV and associated artifacts from the archaeologists and anthropologists. We were instrumental in coming to Honokahua to stop the further unearthing of the kupuna, and were able to establish the burial laws. But he was looking at asking Dr. Fox and sharing how, appreciating how he roamed the Smithsonian and thanked his mom. But for you, Dr. Dowsett, for carrying on the good works of our own Kauka parentage as well. And he was really appreciative of, what we're doing. And his ending quote was La'a Ma'a Pa'a. Anyway, so he's monitoring this and further kind of work that we do. And we appreciate that kind of like feedback. So I don't know. There was other feedback from the audience. JoAnn, do you want to go through it?
Sure. I quickly, before we go into our introduction, I just wanted to share the feedback that we've been getting from people who have logged on to the previous sessions. First of all, so much gratitude to our speakers and our scientists because they really appreciate how the information's been presented and easy to understand for a lot of us lay folks. And I think they clearly recognize your passion in this area, which you've chosen to, build your careers and do your studies. And so much aloha for, our folks. Some of the insights they shared was that they really were surprised to learn about the things like bone harvesting and how big pharma is mining information for new drugs. I mean, that's just not something they hear about all the time. They also said that it provided a really valuable context to better understand some of the systemic racism that's going on in this area. The thing that I was really excited about, because it was really our intention in doing the webinars, was to start the conversation and open it back up and raise awareness. And so the webinars have generated so many good questions. And I wanted to just share some of that with you. Those of you who didn't log into all the webinars, our speakers have addressed some of them. And I know they're going to address some of them in their current presentations tonight. But these are some of the questions that came up. What does advocating for Native rights and sovereignty as it pertains to genetic information look like in clinical practices for geneticists, genetic counselors, and genetic professionals? So here's somebody reaching out and says, what does that mean for us when we're sitting down with clients? They wanted to hear more about the informed consent process in genomics research, including things like long-term storage and use of data. There were many that said, how can I volunteer? How do I spread the word? Give me action steps. Do we sign petitions? Is there Indigenous research that we can and should be supporting? Another said, what's the dream scenario for a Kanaka-run biobank? What kinds of expertise are going to be needed? Yeah, really. Another one, logical next steps for educating the Lahui on this topic. What entity should be making decisions about Kanaka Maoli participation in genetic sequencing and biobanking? So who gets or who should have control of this? This person also said, I have strong feelings about my genetic information. I don't want it in a database. But what if someone in my family participates? What can I do? The other is, how does Leah and Keolu's work relate to each other? So those of you who may be coming in for the first time, we have both a clinician on the front line of patient care, and we have a scientist, bench researcher. So how do they relate? And they do, and they happen to be great friends and colleagues. So the other is, what diseases are Kanaka more genetically predisposed to have? Also, how can, someone said, how can non-native genetic professionals and researchers be allies to the cause without unintentionally stepping into a biocolonialist role? Thank you for that question. After we had session 2, additional questions were, they wanted to hear about models that work. And also, how can we start involving families in the process of granting or denying consent for any testing done on these kupuna or ancient genomics. Are there research grants or pressures that are put on genomic professionals to advocate certain positions or policies within their institution? So are there people who are getting incentives, both professionally or maybe beyond, to market and prescribe genomic tests to patients? And is there realistically a way to abstain from participating in genomic research, or are we beyond that point? So now the task is thinking about how to mitigate the bad impacts and take or make proactive steps, policies, or technology work for us. When we talk about consent, shouldn't the privilege of abstaining from the research always be on the table? Is this still an available option? I know we are going to be covering informed consent, so I'm so grateful for that question. So with all of that background, just so you know that this is the kind of interest that it has created, and Emmet and I are so grateful that it has. That was really the point. I know that we've gotten so much encouragement from our speakers about opening up this conversation again, especially for this topic. So I want to start with introductions so that you can get to the good stuff. Go ahead, Emmet, you can introduce.
Leah. So
Yeah.
Leah, Keolu Dowsett, Assistant Clinical Professor of Pediatrics on the John A. Bird School of Medicine at the University of Hawaii, and a clinical geneticist at the Hawaii Community Genetics, board certified in pediatrics and medical genetics and genomics. Clinical focus includes general genetics and dysmorphology and inborn errors of metabolism. Her primary research focuses on improving genomic health disparities, Native Hawaiian health, and expanding the availability of sequencing and phenotype information to her patients and families. So we're also so proud to have her serve on our Kauka board. Congrats.
I want to introduce Dr. Keolu Fox. His research program is multidisciplinary in nature, reflecting his interdisciplinary research experience in anthropology, genomics, and computer science. Keolu explores the links between human genetic variation and disease in underrepresented populations. Postdoctoral, no, not postdoctoral, he's a professor, assistant professor at the University of California, San Diego School of Medicine. He advocates for more inclusive and representative genomic sequencing to allow indigenous populations to gather and analyze their own genetic data. Keolu's current project focuses on using genome editing technologies to investigate the molecular events involved in chronic inflammatory states, resulting in obesity and catecholamine resistance. Did I do that right? Slaughtering this. I just need you to know these guys are so awesome, and we are just murdering their bios, but you will see what they bring to the table, and not only their education, their insights, their passion, but so much aloha for this, and we really are so fortunate to have them. So I am going to turn it over to you, Leah. I think you're starting the session.
Yeah, definitely. Let me get my slides queued up.
Okay,
Thank you
great.
so much for those introductions. That was awesome. Let me share. And then, is it? No. Yeah, is it going?
We just have to stick.
Good. Looks good, Leah. Full screen.
Like that?
There.
Oh, perfect. Yay. Okay, good. Glad. Thank you, guys. So thank you again. I wanted to, of course, as always, big mahalo to those who have given us the support, like Papa Ola Lōkahi, Ahahui o Kauka, and Ahakane. I also would like to acknowledge Hawaii Community Genetics. It's the place that I work, and I love my colleagues and some of the clinical stuff that I'm going to be presenting today. Really, none of it would be possible without my team. So JoAnn did a beautiful recap of these things, what we kind of covered in the first and second session. So I'll move on. Briefly, just touching upon the cookbook analogy one last time. We know that our genome is kind of like our entire cookbook of DNA, and it's broken up into chromosomes. And of course, the individual recipes represent the genes. And it's when we look at the DNA for spelling changes in the instructions, right, in those recipes, that kind of helps us better understand human disease and kind of leads us to the question, what does that mean for patient care? When we're talking about genetic variant classification, I want to, talk about the word variant, which basically means that there is a change. There is a difference in the patient's DNA sequence that is different from that of the reference. The difference, just because it's a variant, doesn't necessarily mean that it's disease causing. You can certainly have changes that are benign, and we'll talk a little bit more about that later. To review, we all have changes in our genome. You do, I know I certainly do. But what's impactful is when these changes are in critical regions of our genome, right? Like an exon, one of those protein encoding regions that then could lead to disease. So we have this governing body, the American College of Medical Genetics, that classifies, differences on a 5-point scale, varying from benign to pathogenic. And so a lot of factors go into this complex thing that we call genomics, and then translating these results of a genetic test into something meaningful for the patient. Quite frankly, it's a miracle when you begin to think about all the pieces that are involved in this process. And so, as you all know, we are only human, and sometimes we do get it wrong. So what I'd like to do is begin with this cautionary tale. So I'm going to walk through this article that was in the New England Journal of Medicine from 2016, and it was entitled Genetic Misdiagnoses and the Potential for Health Disparities. So this group looked at publicly accessible sequence data from various sources and went back and looked specifically at patients who had a condition called Hypertrophic Cardiomyopathy, or HCM, right, a thickened heart muscle. And they looked at genetic testing in that lab, in that repository, over a 10-year period. When they looked back at their demographic info, they found that a vast majority were, white Americans, and a very, very small proportion, about 8%, were African Americans. And they looked at different variants, right, changes that were thought to be pathogenic, that were thought to be disease causing. But 10 years later, when they restudied those exact same, spelling changes, they went back to the original medical literature. They had taken more diversity into account. They actually found that they had misclassified a lot of these benign or non-disease causing variants as pathogenic. And so what is HCM, or hypertrophic cardiomyopathy? I chose this example because it's pretty common. And when we think about the group of diseases that affect the myocardium, or the heart muscle, we're talking about thickening that can lead to mechanical and or electrical dysfunction in the heart, which is quite serious. Men and women are equally affected. It can happen in children, it can happen in adults. There is no known racial predilection, and the presentation is variable. So someone with a heart that's, a centimeter extra too thick, right, can be completely asymptomatic, while another person with the exact same thickness in their heart ultrasound measurement can have severe heart failure. Some patients, unfortunately, also present with sudden cardiac death. So being that it is pretty well studied, I think it's a great example of how we've been kind of following the data over time. We've been studying cardiomyopathy genetics since the 90s, and since the 2000s, we've been using genetic testing to kind of guide how we take care of these patients, right? So the idea being that a negative genetic test or a screen would eliminate the need to do surveillance for these patients, right? Like annual, heart ultrasounds and things like that. When we think about the classification of heart disease, again, when we're specifically talking about genetic testing, we're trying to identify primary causes. So these are things that are intrinsic to the myocardium itself, right? We are dealing with processes that are like kind of confined just to the heart muscle. There are a lot of secondary reasons by which we could have thickened heart muscle. Due to other medical conditions, right? This could be things like coronary artery disease, high blood pressure, autoimmune disease, muscular dystrophy, et cetera, and acquired things that could be, exposure to chemotherapy that would cause thickening of the heart muscle, long-term chronic exposure to illicit drugs or alcohol, different types of viral infections, and pregnancy itself can also cause it transiently. So when we as clinicians are looking at a patient, right, we look at their history. We ask them questions. We want to know about the physical findings in this patient. Is this patient obese? Is she an athlete? Does he have diabetes? Can patients have different experiences? Certainly. We ask questions like shortness of breath. Are you experiencing palpitations, fainting episodes, chest pain? All of these things help us as clinicians in terms of our exam. We, of course, pay attention to the heart. Every checkup you go to, right? We listen, we check pulses, we palpate, we feel the chest, but we also pay special attention to imaging data too, right? We look at heart tracings, the ultrasound of the heart, and all of that together, in addition to the family history, helps us get a more complete clinical picture. When we think about doing genetic testing, at least in my office, right, we're again hoping that by doing genetic screening, we can decide whether or not we can do a very serious or a less serious path for surveillance. And so if we are looking at 100 genes at once and we actually find a hit or find a reason for why you have a thickened heart muscle, we can know more about the reason why that's happening in your body through the genetics, and then think about specific kinds of treatment. Of course, lifestyle adaptations are important regardless of your genetic testing, right? We always try to minimize risk. With healthy exercise and diet things. We recommend certain types of monitoring. We also might have recommendations about medicines or implantable devices, right? If your heart is so thick that we're worried about the rhythm, maybe we need something like a pacemaker, right? Which can be life-saving. So in this paper, they found that Black Americans with respect to two specific variants were disproportionately misclassified. And again, they were told that they had this disease-causing gene, when in fact, after taking into account better diversity and people, with genomes from the African continent, they were able to kind of figure out that actually that's just a change that's very, very common in people of African ancestry. So what happens? Well, if you have a finding that is pathogenic on your genetics results report, you might end up getting a defibrillator implanted, and then 10 years later find out that, I'm sorry, that was an incorrect diagnosis. And there are far-reaching effects beyond the social and economic implications of that when you have an incorrect diagnosis. But also there's false reassurance, right? For these people who had the genetic test and were cleared, now you have to go back and tell them that what we tested for was actually completely false. And so what did we learn from this study? Moving forward, we kind of want to consider diverse populations, because if they had done that in the first place, they probably would have avoided this misclassification altogether. But also too, additionally, right, when we think about people from smaller or minority backgrounds, we expect that these changes, right, these variants are going to be reclassified or upgraded or downgraded as more testing increases, right, as testing becomes more available to other people. And so, how do we go about doing this? How do we create uniformity and consistency? And that's something that we'll talk about later on. Historically, in underrepresented minorities, right, in URMs, we have lower diagnostic rates when it comes to genetic testing. Sometimes this has to do with phenotyping. So what does that mean? As a geneticist, when I'm looking at a patient for a specific syndrome or a condition, I try to make sure that we're using clues, right, from their physical appearance or medical data to help filter out what we find in the testing. And the second piece of that, right, is databases. A lot of times we have this predominance of Caucasians being represented in the medical literature and these databases. And again, the result is that the outcome can be different for different ancestry groups. I want everyone to just be a little bit familiar with this database, this exact database. It came onto the scene in 2013, and the MacArthur Lab, right, which is under now this division of the Broad Institute, really kind of changed the way we kind of looked at genetic information. Looking at population data like never before, right? They had this database that had over 60,000 individuals that allowed us to kind of get a sense of what's normal and what's abnormal on, even in the rare end of the spectrum. And so just to give you guys a flavor, when we highlight the underrepresented minorities that were in this database that came out, about 6-7 years ago, about 40% out of the 24,000 samples were URM which is, I think, comparatively for that time, pretty impressive. Prior to that, like Keolu had talked about in one of his previous, sessions, the genome-wide association studies that kind of summarize all the different publications between 2015 and 2018, a vast majority, like something like 90% were Caucasian and only 10% underrepresented minority. So getting up to 40% was kind of a big deal. But over time, as they added more and more participants right now, I mean, back in 2016, close to 250,000. And again, we're still expanding. The proportion of underrepresented minorities has stayed the same. And I think we, as a scientific community, can do better. And there are certain initiatives that we will also talk about shortly that are wanting to begin to address the current gaps that exist for our minorities. Where I work, we kind of wanted to look at the effectiveness of genetic testing, a specific test called whole exome sequencing, and how that might be different based on ancestry group. And so we divided our results into 3 little buckets: positive, uncertain, and negative results. Really quickly, whole exome sequencing. So what is that? When we think about our entire genome, right, we have about 20,000 or so genes that are known to be important instructions for growth and development. And exome is the most comprehensive genetic test that is available clinically. There is genome sequencing, and we use it very rarely in clinical situations, but I would say exome is certainly more widely available. It's utilized as a diagnostic tool when we are trying to identify genetic cause for someone's symptoms, right, or someone's disease, but has previously not been identified. And the reason why we like to focus on the exome is because more than 85% of the genetic changes that cause disease are found in the exome, which is still only 1 to 2% of the whole genome. So it's really just a high yield test. It's not a perfect test. So we know that there are things that are missing, including, imprinting causes, triplet repeat disorders like Fragile X or Huntington's, mitochondrial disease that cannot be picked up by this test. So again, no test is perfect. But what I wanted to impress on you guys is just what happens from the time you give your sample to getting this genomic data and how that then gets back to you in a result or a report. There's a lot of different analyses that need to happen. And again, we use those phenotypes or those clues from the patient itself to kind of filter out these variants. And so when we see changes in somebody's DNA, we have to ask the question, does it disrupt gene function? Is that disruption, is that going to lead to disease downstream? And then after all that is said and done, if we've proved those things, we also have to then tie it back to the patient and make sure that, does this make sense for this person? When we think about a sample going through the machine and getting genomic results, we can actually have over 100,000 variants or changes from one sample alone. So to get an answer, it takes a lot of time and it takes a lot of filtering, right? A lot of PhDs and MDs and genetic counselors and lab techs and bioinformaticists are kind of putting their minds together to sort this all out. The NIH has an undiagnosed disease program, and they do genome and exome sequencing for underrepresented minorities too. Their hit rate is only about 20%. So again, we're approaching national averages, but again, across the board, whether it's locally or on a national initiative, we know that minorities have smaller positivity rates. When we're doing informed consent for a test like whole exome sequencing, we have to kind of explain a lot of things. Of course, to most people, genomics is not in their everyday vernacular. And for us, we want to really make sure that our families understand exactly what this test entails, because it is such a big test. Informed consent, includes explaining what the test is. It has to cover all the different possibilities for results, like positive, negative, or uncertain ones. We also have to disclose that a test as big and broad as this one sometimes can show some unexpected biologic relationships, like non-paternity as an example. Sometimes we have information about family members that can be inferred. let's say we have a very, very sick baby in the ICU and, the baby is having seizures from the, day one of life. We are maybe thinking about this very big, broad test to figure out how come baby's having seizures. But because we're looking at 20,000 things, we may also uncover, that there is a disease-causing finding in the breast cancer gene, right? So that might infer that mom or dad could be at risk, right? Because they probably also harbor this change that puts them at increased risk for hereditary cancer. And so what is that supposed to mean for a family? And do they want to know that kind of information? We also ask about, uncertainty. And sometimes we use both maternal and paternal lineages to help with unclear results. And so for people, no worries. When we have somebody who, is adopted or comes from a single parent home, we may not always be able to resolve that uncertainty. We have to talk about what the test doesn't include. We also have to talk about results being dynamic, meaning over time, right? Like the study we just went through. We get more data and things can change. What is pathogenic could be benign, or what's uncertain may be also upgraded to being disease causing. Samples may be kept by the testing lab for research purposes. All of that we need to talk about the cost, right? Does insurance cover this? What might your out-of-pocket costs be? And then, of course, very importantly, if we were to find something, are you going to be discriminated against because this condition might preclude you from, getting disability insurance or something like that down the road? When we're thinking about informed consent for research, we're talking about an agreement, right, between the researcher and the human subject, where we're thinking about, of course, participation. And with that, the researcher kind of agrees to do all these things. I borrowed this from Chang and Lowenthal, and this particular paper discusses at length, some of the vulnerability to Native Hawaiians with genetic research. And so, of course, describing the research, talking about risks and harms, right? Things that, might be other procedures or treatments that might be alternatively sought out instead of, participating in the study. What happens if an injury occurs? What kind of compensation is there? We have to make sure that we prove that this is voluntary, right? We want, if we're in a, community-based health center recruiting patients for different types of studies, we want people to know that this is completely voluntary. And if you choose to decline from being a part of this study, there's no threat, right, to the quality of your healthcare from there on forward. Redoing informed consent as things evolve, how do we really kind of address that as a whole? And so looking back at our group, and specifically when I see patients, we looked at one calendar year and we had done 72 exomes, or 72 of these big fancy tests. Of that, 65 of our patients were from underrepresented minority families, 7 of whom were Caucasian. And this is kind of like the more specific breakdown. We had a lot of Pacific Islanders, but, like most of Hawaii, cosmopolitan, right? Plenty of people are of mixed ancestry. So this is a typical pedigree that we see in our clinic all the time. The patient right there is a 15-year-old male. Squares are boys, girls are circles. And you can see that on the father's side of the family, he's Japanese, Hawaiian, Portuguese, Irish. On the other side, on mom's side, she's Japanese, Hawaiian, Chinese, Scottish, English. And again, lots of admixture here in Hawaii. And what that leads to is some differences in our ability to kind of give meaningful information back to our families when we're talking about genetic testing. So the first finding that we did kind of look at after taking a year of patients and separating it out was that the diagnostic rates in Caucasians is much, much higher than those who are of minority background. The uncertainty level is actually the same, which was a surprise to us. But what was really poignant was that for our underrepresented minority families, we have to sit there, look them in the eye and explain to them that even with the best test that we've got, we are unable to find an answer 42% of the time. For our Caucasian families, we never had to do this. And I think, again, we're not getting the same quality of answers between groups. So all of us is this initiative, right, that NIH is moving forward with, and it is a precision medicine initiative. So what does that mean exactly? They want to, include your data as an individual into this massive database, and they want to kind of look at the interaction between environment and lifestyle, and of course your DNA, and they want to make customized recommendations for people of different backgrounds and ages. And again, the goal is to overall reduce costs by matching the right person with the right treatment at the right time. That all sounds great. And again, they are focusing on building a more diverse database. So what does that mean? They actually want over 1 million samples from people from minority backgrounds. Again, they want to figure out what treatments work best for people of different backgrounds. So Keolu is going to talk about that. We also know that there are different risk factors for certain diseases that we need to kind of pay attention to. We want to connect people with the right clinical studies, and learning how technology can help us be healthier is, again, a big goal of this effort. And so on one hand, you know, right, that leaving people out is an equity and a knowledge issue. To make new genetic discoveries and to have true knowledge of genetic variation, we need to include people of diverse backgrounds. And we need to include them in genetic research at all levels, not just as participants, but on the research and the principal investigator levels alike. By increasing participation in genomics research, and in the databases, the capacity for social benefit certainly increases. On the flip side of that coin, we also know that minority communities have a good reason for being wary of medical researchers, right? With lack of accountability, lack of trust that has been demonstrated time and time again. And so, Indigenous communities certainly want to experience direct benefits, right, from participation and collaboration in genomic research, right? We want to benefit, but how can we achieve this? I, for one, ask myself on a daily basis, how can we do better to translate the complexities of genomics into actionable healthcare endeavors for my patients, right? How do I take better care of my families? And, with that in mind, I want to transition over to Keolu, who's going to talk a little bit more about sharing power and enhance engagement in research and control over data sharing, and what that might look like for Hawaiians.
Right on.
Okay, I think I have to stop share, sorry. Okay. Yeah.
Okay. Ready? All right. Aloha, everyone. Can you hear me?
We can. Thank you.
Oh, okay. Good. I had to put my glasses so you guys take me serious, So, but lovely to see everyone. Thank you for the kind introduction always. So let me share here.
Did I stop share? I hope so.
Oh, can you guys see my thing? Oh, how about now? Share. Host disabled participant screen sharing. Oh, somebody has to make me the sharer.
Okay, I need to call in our technical team and help us get
Oh, now it's all good.
access.
I got you.
Thank you, everyone.
Raja. Okay.
Good, I can.
All right. And I have a lot to cover. So, and I want to do this justice because it's complicated and it's very important. And it's an honor to get to talk to everyone about these things and have the kind introductions, and then have my wonderful partner in crime get to kind of set up a lot of the pieces that we're going to talk about. And first, just again, thank you to our community partners. None of this is possible without you and without our participants from the community themselves. And so now I wanted to kind of transition and talk a little bit about emerging tools. So the first part of this, we're going to really focus on rewriting human history and empowering the Hawaiian community with genome editing tools, a little bit about that, just to kind of tune up. And then we're going to slide right into the conversation about data as a resource, data sharing, data privacy, and kind of emerging and futuristic models around that information. So as Leah alluded to, in a lot of ways, there's this inborn bias that has sort of been manifested for a number of reasons within the genomics research community around who's participating and has been included and engaged in the future of predictive and preventative medicine. Now we're starting to get a little bit familiar with the different types of techniques and technologies that we use to survey the human genome in an attempt to look at human genetic variation. So we heard about exome sequencing, we heard about whole genome sequencing, we heard about genome-wide association studies, and this slide has kind of a breakdown of the participants that have been included in these sort of just on a massive scale of genome-wide association studies up to about 2018. And you can see this kind of 88% of 40 million individuals included in GWAS are of European ancestry. And that's highly problematic for a number of reasons, because when we develop algorithms to model and predict and prevent any type of common complex disease or rare diseases or unknown diseases, we don't have the same accuracy with some individuals as we do with others. On top of that, the reference genome that we use to compare every single genome that's sequenced to is an individual of Western European ancestry. So that is to say, there is this inborn, baked in bias in the whole field itself. And we're gonna think about and talk about what the consequences of that are. And we're also, both historically and clinically, and the way we understand history and the way people narrativize data when they sell it to a journal, right? Not like sell it for money, but when they sell a story to reviewers at a high quality medical journal, there is a whole element of talking story that goes into that. And we're going to talk about how that works and how people have narrativized data. And then we'll finally wrap it up by talking about solutions, because solutions are the way forward for our communities. So these are data from today. Literally right before this talk, the reason why I was late was because I was bringing this data down from a database that's sponsored by the Oxford University, someone by the name of Melinda Mills, who does good work over there. And you can see that today at this time, West Coast time, 6:04, we can see that 87.46% of these genome-wide association studies have exclusively featured individuals of European ancestry. That's hot off the press. And then you can see here's other. This is us, everybody else in the Hawaiian community, okay? 3.09% mixed ancestry. That would be pretty much everybody here. So that is, again, highly problematic. Now, something that's very, very interesting that we haven't quite talked about yet, I think we have dialed in a little bit, is that 72% of the genetic discoveries have taken place in exactly 3 countries: the UK, the United States of America, and Iceland. Funny enough, Iceland does a lot of work within their own community and for other communities. But one of the things that's interesting about that is that means that those clinicians and those doctors are the ones that are and scientists like myself, those are the individuals that are interpreting that data. So if you know that 72% of the data generated comes from 3 countries, that means that the medical doctors and scientists within those institutions also come from or participate in scientific and medical systems within those 3 countries. And that's really, really problematic for a number of reasons, because it means that the people interpreting that data sometimes aren't from the countries themselves that those individuals might come from. So that can lead to the development of something called just so stories. And this is a term that has been popularized by evolutionary theorists, namely these 2 guys, Stephen Jay Gould and Richard Lewontin. And what they did is they based it off of this child's story called Just So Stories. And you can see this picture here. This is by this guy, Roger Kipling. He also wrote, maybe you're familiar with the Disney movie, The Jungle Book. And he wrote this book for his daughter, actually. And what you see here is an elephant getting its nose tugged on, its trunk tugged on by a crocodile. And so the narrative is that the reason they have long noses is because crocodiles pulled on them for a really long time. Now we know that's not true. We know that's a child's story, but this is called a just so story. And this idea has been used to popularize all kinds of evolutionary explanations of data in genetics. And these are called adaptionist narratives. And I want to explain a few of them to you. So some of the things that I work on in my laboratory that are very interesting. One of them is, how has the environment shaped our genomes over time? And I think we talked about this a little bit last time in our first lecture, but we never really got to kind of dig into some of these stories. One of them that we know of is that individuals who live at high elevation in the Himalayas, where they've been continuously living in really, really high places, have adapted to high altitude over time. And you can see this beautiful image of this individual looking over at some of the tallest mountains in the world. We know actually Mauna Kea is the tallest mountain in the world, but no big deal. But then we have another story that has been interpreted, and this is Indonesian divers that have evolved bigger spleens to hunt underwater. So two of these are evolutionary stories where it's like the environment and the Aina itself has impacted and shaped the genomes of these communities. Now, from the medical point of view, if you're not from a high elevation community and you're carrying a child, there's a really high chance that you'll have a miscarriage if you're a woman, because your genome is just not shaped for that type of subsistence at that high altitude, right? Lots of people have to carry oxygen when they hike up different mountains and so on and so forth. So my question for our audience is, and I want you to think deeply about this before you make your responses in the chat. Which of these narratives is true? I just spun up and talked story like we do, and I told you two different stories. What has more of a profound impact on the genome? Chronic hypoxia, because of 8,000 years of subsistence at that elevation, or acute hypoxia, repetitive diving. And for all of everyone here that likes to free dive, you know there's some people who can hold their breath for a really long time. So which ones do we think are true? Does anybody want to put something in the chat or say something, or anybody want to chime in? Or maybe Leah or somebody can read it for me, whatever people say. Anything? Okay, well, I'll tell you anyway. What it looks like, though, is that there's probably a much higher chance that people that have chronic hypoxia, that have been experienced chronic hypoxia, are going to have genetic mutations in certain genes that influence their genome. However, there is this whole story that has manifested about acute hypoxia. Now, this was done by investigators that collected DNA information in this Baju population in Indonesia. And not only is it scientifically inaccurate and it doesn't have sort of mechanistic evidence to support it, it also was accused of being helicopter science, helicopter research. So they got accused in Science magazine of did a study of Indonesian people who spend most of their days underwater violate ethical rules. And so it's one thing to have an ethical violation because you weren't supposed to take DNA out of that country and narrativize it somewhere else, but it's another thing to create a scientific narrative that's incorrect, that defines someone else's history and is not based in actual biology or medicine. And so we see that there can be these potential points of conflict and friction in the way that people identify mutations that look like they're quote unquote under natural selection and the way that we create stories about them. So here's a whole map of many of these different stories. Some of my favorites are blood group mutations, and I kind of can explain that in a little bit. And at the top of the talk, JoAnn kind of told you that there are mutations that they're globally rare, but amongst individual populations, they're quite common. And so you can see that our ABO variation that we know of, in South America, I've worked in South America, over here, In Brazil, in the Amazon, they don't even stock any other unit of blood other than O blood, because they know everybody's going to be O, because of it's almost reached fixation, the O blood type. And that is a result of something called balancing selection. And so we know that there's these forces that have shaped our genomes, but nothing has been more profound in shaping our genomes than pathogens. And we are currently undergoing something like that right now with COVID-19. But we can look at HBB. The HBB mutation is responsible for sickle cell anemia, and that's a result of equatorial populations in Africa's interactions with the malaria parasite. There are, here are these high elevation adaptation mutations in genes involved in something called the Hif pathway. And one of the famous ones is EPAS1, and it's been involved and identified in 2 different populations at high elevation. One of them is Tibetan people, high elevation Highlanders, and Nepalese. And then they found a similar mutation in the gene EGLN1 in the Andes, where there are other high elevation populations. All of these populations are indigenous people, right? All of their mutations have been identified in those populations and then brought to America or London or something like this to be narrativized and interpreted. There's another one that was discovered in the UPIC that has to do with metabolizing different types of marine fat over time. So you get the point. We identify mutations that look like they're under selection, and then we create stories about why they exist, why they're there, why they're at a high elevation. So why am I telling you this? Because I think there's a narrative that exists also, just to get meta with you, that medicine and evolution are 2 different things, but they're not. They're the exact same thing. They contribute to the way we contract diseases over time, including chronic complex diseases like cancer and heart disease, the #1 and #2 cause of death in the United States of America. So it gets you thinking about the way we create and spin stories. So now, how is this relevant to Polynesian and Hawaiian people? Well, let me tell you. This mutation was discovered in a gene first described, and looks like under selection that it's in Polynesia, first described in Samoa. And the investigators who just described it are not of Polynesian ancestry. And the story they created is very, very interesting. And the reason I'm talking about this is because Uncle Emmet mentioned this idea of the Thrifty gene theory. So it inspired me to talk about this individual narrative. And so the Thrifty genotype hypothesis is something that originated in the 1950s by a guy at the University of Michigan named James Neal. Now, this is in the back end of when a lot of scholars, quote unquote scholars, thought that the Polynesian or Austronesian migration had occurred from the wrong direction. They thought that, and they tried to popularize this idea, remember Thor Heyerdahl, that our ancestors, our kupuna, came from South America. And we know now that's not what happened. But in light of that idea about accidental voyaging and all of these other things, the Thrifty gene theory was invented. And that's this idea that there were genetic mutations that would predispose you to storing calories. So something called hypercaloric storage. So maybe during a voyage, you would have, you would oscillate between feast and famine and this active lifestyle and a lifestyle where you didn't have any food on the boat. And then as we move into a more modern lifestyle, those mutations that were really important or advantageous at that time predispose you to type 2 diabetes or obesity. And that's basically what this idea of the Thrifty gene or the Thrifty genotype hypothesis is. And investigators in 2015 decided to reinvent it and bring it back to life, even though it had kind of been disproved for a number of reasons. And what they said is they observed mutations in this gene, CREBRF, that was found and first identified in Samoa, who are obese. But the most interesting thing about this was that, and anyone who's ever done a rotation in endocrinology can vouch for this, but there is, they identified these Thrifty point mutations in this gene that predispose to obesity, but protect against type 2 diabetes. Now that's crazy for a number of reasons, because whenever you expect to see a linear correlation between type 2 diabetes and obesity. So as one goes up, so does the other. That's what we expect to see. We don't expect to see a protective genetic variant against type 2 diabetes, and certainly not one that its origin would have been through thrifty means. So their story to me seemed convoluted in 2015. It seemed like it wasn't rooted in a complete constructive understanding of our history. And their just so claim in this case was that the selective force that maintained this mutation in Polynesia was related to a scarcity of calories during our diaspora. And I'm happy to answer questions about that later, but obviously the media loves to run away with stuff like this. So you have the Japan Times talking about the gene that may benefit sumo giants and how a powerful obesity gene helps Samoans conquer the Pacific, and all of these kind of harmful narratives about the innateness of obesity and type 2 diabetes that is related to our diaspora. Now, we all know that we're the greatest voyaging community of all time. We all know that we use complex metadata like bird migration patterns and wind and weather patterns, and we really got busy at night when we used the celestial sphere to find our way. And anyone who's ever spent time at sea knows this, and you know how complex it is. And you also know that we had plenty of different types of canoe plants and pua'a and other types of protein and catching fish and all of these things that weren't consistent. So what it says to me is that this narrativization of a thrifty mutation is about discrediting our accomplishments as a community in our history, and saying that the reasons why we have extremely high rates of obesity in Polynesia is innate, and it's in our genome, and it's not a product of separating us from our hunting rights and our fishing rights and our access to the moana and our access to the reef and a bunch of other cultural things that are really the result of the residue of colonialism and imperialism. So, but when we look at the top 10 or so countries in the world, you can see that we do have some of the highest rates of obesity in the world in Polynesia. So it played out in the way that the narrative was popularized. So when we look at other Polynesian communities besides Samoa, what do we see when it comes to this thrifty mutation? We see it's quite ubiquitous in Polynesia. We don't see it in any other population. And you look at the NOMAD database that Leah was talking about, and you can see that the mutation is strictly found in island communities. It was most recently identified in the Chamorro population, which is interesting because we've created these divisions between Micronesian and Polynesian people that don't actually exist. And we know that's a source of a lot of conflict in Hawaii and today. But I thought it was very interesting that it is found everywhere from Tahiti to Samoa to Aotearoa to Hawaii. Now, the real question should be, what actually does this mutation do? Because if it's thrifty or not, we need to know what it does mechanistically and what types of technology would you use to investigate that. Now, I love reverse engineering things. Ever since I was a little kid, like if there was a problem with the washer or the dryer, I would fix it. If there was a problem with the car, I would fix it. If there's a problem with the boat. So I do the exact same thing at work now, and I use different tools. What are those tools? I like to use CRISPR-Cas9 genome editing, or a specific type of genome editing called base editing. And what we do with that technology is we can kind of find and replace the typo, and we can bring that mutation into a stem cell and see exactly what it does. And these are called isogenic cell lines. And we can literally reverse engineer a point mutation that is specific to Polynesian people and see what it does in context. We can, and that's exactly what we did. This is just kind of data confirming that. I'd like to say this too, because I'm cheap, yeah? We did this for $500, and most of these companies would charge you 20 to 25 grand. So I'm really proud of that, yeah? But what you can do is introduce it in the right cell line and you can test for insulin sensitivity, you can test for inflammation, you can test for metabolomic or metabolism profiles. And these kinds of systems, these reverse engineering systems, can be easily adapted to gain insight into the role of other genes and their connections to things like obesity or cancer or heart disease. So playing the reverse engineering game can be very valuable for us when we think about not only rewriting our history, but also understanding health and kind of creating specialized and precision forms of treatment. We also got to work very closely with the Moko Foundation and several professional rugby teams in Aotearoa. And what we noticed is that in individuals who are of Polynesian heritage, who have this mutation, it's much higher amongst professional athletes. So our narrative and the story we're making up is that it's the tall, dark, and handsome gene. So, just kind of thinking about what some of the more dark interpretations of this and what people are using genetic information to do, the non-Polynesian scientists who identified this mutation actually filed a patent claim attempting to commercialize the exclusive use of the physiological response to this gene, to this specific mutation that was discovered in Polynesia, which brings up a range of questions around what this means in terms of designing the next generation of type 2 diabetes drugs based on a mutation that was discovered in Polynesia. The immediate questions that I'm thinking are, is our community going to get a cut of the piece of pie? Are we going to get subsidized access to the drugs? Are we going to get a percentage of the intellectual property? Is this going to be like every other extractive process that is kind of connected to resources? And in addition to that, DNA has been integrated into surveillance systems and other things in countries like China, where the Uyghur ethnic group has had their genomes sequenced, and it's not looking good. And a lot of these kinds of technologies are being integrated and embedded with all kinds of things. So it's something for us to think about in terms of who has access to genetic information and what is it used for. Is it embedded into a surveillance state? Is it used to, is it commodified? And how is it commodified? And what happens to that resource? So another question, lasting question that I'd like you guys to think about is, should all genomic data be open to the public? And if so, why? These are deep questions that no one really has the kind of end of the road answer for, but it's something to think about further. I'd also like to connect this historically to the creation of the Human Genome Project and say, look, the Human Genome Project was founded in 2001. I don't even know if I was in high school yet, but I remember learning about it. And you have then President Clinton. At the time, they sequenced one genome. It was the genome reference. And the #1 commodity on planet Earth was oil. And you can see I have that here. Oil equals money bags. Okay. We moved to the Precision Medicine Initiative, or what Leah was introducing as the All of Us program. And you can see that they're attempting to sequence 1 million genomes. And in 2018, you get the kind of transition to what we are calling the digital economy, and the recognition that data itself, raw data, including digital sequence information derived from human genomes, is the #1 commodity on planet Earth. And you can see this article from The Economist magazine, and they're talking about the world's most valuable resources, no longer oil, but data. And the data economy demands a new approach to antitrust rules, and thinking about why the top 5 or 10 companies in the world, including Amazon, Google, Microsoft, Uber, Facebook, etc., all rely on the digital surveillance economy. So genomics fits into that economy, and we should be treating it like a resource. And this book is absolutely excellent. It's called The Age of Surveillance Capitalism. I read it a few years ago. It describes a number of these ideas, but I just wanted to say, like, we should start a book club after this so we can kind of share resources. But this is a really good one that got me really thinking about data as a resource and making comparisons with data to oil and timber and diamonds and other things. The All of Us program, though, hasn't really recognized that. They've kind of moved forward full steam ahead. They've announced that they want to sequence 1 million people's genomes. They've also doubled down on the fact that they want to ensure that at least 50% of those genomes are from underrepresented communities, who we know, if we go back to the top of this talk, because of the unique ways in which Indigenous and other communities have directly interfaced with the Aina for tens of thousands of years, that shapes our genomes. That means that it's going to be a lot easier to identify potentially medically actionable mutations within our populations. And they've already done it. And one of the examples was that CREBRF mutation, right? And that brings us to the emergent field of Indigenous sovereignty. Listen, I'm not the only one that's talking about this. This is a global phenomenon, and we have to work on providing solutions that empower our communities together. So let's talk about that. We put out this paper that JoAnn was really kind enough to share with the community about recognizing Indigenous rights and Indigenous data sovereignty in raw data, and what that means in terms of an open data environment, and different kind of techniques and ideas around sharing digital sequence information and ways to collaborate with potential partners and projects that are consistent with existing protocols. And finally, how to negotiate equitable outcomes for the use of this information, including commercialization. And there's a review on this, which is the first of many new forms of innovation in the data science space. It's been in the news quite a bit, thinking about COVID-19 and how Indigenous communities from Navajo Nation to Lakota to Hawaii and all over the world are thinking about ways to strengthen our title to and our recognition of the owners of our data, and what that means in a world where more genetic information has been collected in the last 8 months than in the entirety of history. Think about how profound that is. Think about how much money Pfizer is going to make in the development of this vaccine. And in the process, how much genetic information was collected every time a genetic test was done with new techniques and things like swab-seq. We have to think about the future. We've always done that as a community, and we won't stop. And there's precedent for designing drugs based on mutations that have already been identified in communities. Regeneron, who you might remember when Trump had COVID, was the company that gave him the antibody booster. That same company is working towards the development of a whole new class of drugs. One of them that was produced is called PESCI9 inhibitors. These are a new class of cholesterol drugs that have already hit the market. And you can expect genomic information being used to expedite the research and development kind of process in the way that blockbuster drugs are developed in the future, because they're already doing it. Another example is Vertex. This is what everyone's calling the $300,000 drug. I believe we briefly talked about this in our first talk, but I feel like I have to bring it up again, because I don't want to live in a country or any type of society where we have a broken healthcare system, where almost 30 million people don't have access to healthcare, and genetic information from patients that have cystic fibrosis is used to develop drugs that are sold back to that very same community for $300,000 a year. We have some serious issues around access and the development of drugs, and we need to recognize that. So, given all of that, do you support the NIH's All of Us program? If so, why? These are questions that I want all of you guys to think about, because in the end, it's your genome, it's your data, and you have a personal trust and a potential to participate in these studies, and you need to understand and think about, like, what are the potential impacts that this is going to have for you and your ohana and the Hawaiian community broadly. And that brings us to some solutions and some ideas that are motivated by the Nagoya Protocol's you know, these ideas focused on access and benefit sharing. And what does benefit sharing mean? Well, benefit sharing has to do with control and access, and basically ensuring that you and our community receive portions of the royalties that are developed from various types of drugs that are derived from genetic information. And benefit sharing is the action of giving a tiny portion of advantages or profits derived from the use of human genetic resources to the resource providers. And there's precedent for this in agriculture, big time. Rooibos tea profits are being shared with indigenous communities in Southern Africa. And benefit sharing has been transformative in Botswana, in Africa, where diamond mining has completely transformed the economy in a way where 5% of royalties from diamonds go directly into cultural revitalization programs and programs around health and education. It's been pretty cool to watch, but you can see why we're making these comparisons between other industries where resources are being extracted and benefit sharing models that have already been applied. I talked about this in the New England Journal of Medicine. Really proud to get to debut and talk about a lot of the strategies and potential solutions that we can think about with respect to this. And I actually got to respond to those individuals in a direct letter. And in that letter, I said, genomic data are a resource. And I said, I envision a future in which the innovations of precision medicine benefit the communities that made that innovation possible. Will need more than diverse data sets to get there. And that's the bottom line. And I think in the future, we have to explore models where we understand and find ways to secure our data. One of those ideas is called data trusts. And the Atlantic Council is a really interesting data-oriented think tank where they are thinking about data as a resource and understanding that data trusts, whether it's our community broadly, your family, or an individual data trust that represents you alone, allows you to have control of your data. We are talking to a number of different stakeholders from the top-down level, working with the World Economic Forum, the UN's Declaration on the Rights of Indigenous People, the Atlantic Council, and the UN's Big Data and Artificial Intelligence Global Pulse on thinking about ways to recognize Indigenous data as a resource. Some people talk about it, we're doing it, and a lot of our conversations are centering around Indigenous data sovereignty and data trusts. So what's the future of genomics? Well, one is recognizing and think about ethical genomic research with Indigenous communities. There's no substitute for grassroots research at all. And this is from a meeting on metabolic health in Aotearoa and Ahipara. Here's me in the back. And we are working on many of those different ideas. Sorry about the dog, yeah. We're always thinking about democratizing technology. The more people who have fluency in understanding technology, the better. Here's my friend Hayden Henry in Aotearoa. He is just a testament to the whole like one size does not fit all model of the way that we're treating science. And you can see Uncle Hayden is doing his first PCR here. We've had the Singh Indigenous Genomics Conference, and it was very successful. This was the first International Indigenous Genomics Conference with all Indigenous speakers. We would love to have a Singh Hawaii where we're activating the next generation of Kanaka genome scientists, hoping Leah and everyone here can be involved in the leadership of that initiative in the future. We're really focused on algorithm fairness and understanding ways that the future of algorithm development will be applicable to reducing bias in medical genomics. We put out a paper with my friends from Howard University, and it's all focusing on including vulnerable populations in the assessment of data from vulnerable populations. And I was inspired by something that that Nainoa Thompson said at a talk that that I saw in Honolulu last year or the year before, and he was talking about remapping the Pacific Ocean. And I feel like that is exactly what we need to do as a community with respect to genomics. We need to remap the human genome. We need to think about the many different ways in which we create a Hawaiian genome reference that we use to impute and infer and predict what mutations do with respect to our communities so that we can have better access to treatment. And an example of this was a Melanesian investigator and a bunch of collaborators there created a Melanesian specific population genome reference, and they were able to identify new human genes that had never been discovered before in 2019. So I'm trying to explain how biased this human genome reference is currently by saying, we don't even know how many human genes are out there yet. We have no idea what exists. And we've been basing discovery on one population of people. So what we do in the future is going to be very exciting. And I think that represents opportunities for solidarity within oceanic genomics. I put the Star of Oceania from Kawika Kalakaua and this He'e map of Polynesia, kind of connecting all of our communities and recognizing the opportunities to create a Polynesian genome reference in the future and the impact that will have. And we're working on this with my partner, Dr. Hinano Murphy, and thinking about the Indigenous Futures Lab Field School in many of the projects that we're hoping to develop in the future. And thank you. Take your questions. Okay, all good? Sorry, a little long, huh? Okay. Oh, I can't hear you, JoAnn, because you're muted.
Thanks. I did want to remind you on time because that was just so fascinating. And we did have a couple comments real quickly. One comment was that when you talked about the vaccine or the one comment said that genetic technology also allowed Pfizer and Moderna, the lots of others, to develop a COVID vaccine in 9 months rather than 10 years. So yes, we've all been following this fast track. So as a health educator, I always think who's going to have access to the vaccine. So that's going to be an issue. But somebody also said they were asked for ethnicity during their COVID test and wanted to know if that information could be sold or would be sold.
Good question.
Great question. Yeah, I mean, you know, depending on, I mean, I'm assuming that the test was probably done for, you know, like on a clinical basis, meaning, you know, when you're thinking about taking a flight or whatever and you have to take the test, or if you're symptomatic and your primary care physician wants to know, and then you just went to like a routine lab to take care of that test. I'm surprised to learn that they are asking for you to self-identify your ancestry, but at the same time, you know, I think it would be wrong, and I don't believe that, you know, at least a lot of the labs here locally would sell that information to another company. You know, we do know that, of course, right, minorities like ourselves are more infected because we're more exposed and less protected. And I think, you know, one of the issues with the COVID-19 trials, right, is it lacks minority representation, especially when we're the ones that are most impacted, right, by COVID-19. So perhaps that's why they were asking, more so just to know the demographic from that aspect, but yeah, not for purposes of like, you know, selling your genomic data.
Well, I also know early on in the lockdown, one of the things that, and Emmet and I were in conversation with Office of Hawaiian Affairs, is they wanted data. They wanted to know who was being screened by ethnic group. We were concerned that a lot of the data coming nationally was AAPI rubric again, which is not helpful. So, you know, one of the suggestions was they should be asked their ethnicity when they're getting screened. So interesting, then the question comes up, what do they do with these tests and this information, knowing that there's so many possibilities? Emmet, do you have any comments?
Yeah, So it's a couple of things people want to know how genomics apply to COVID-19. That's so exciting. It's not only the Native Hawaiian, Pacific Island, Filipino kind of like statistics all over. It's now impacting the Micronesian and also Taiwan and Samoa and Vanuatu. And so people want to see what it is that has sustained us or have influenced us with people coming together. And so if there's any kind of idea about how to do that, that would be kind of like an interesting follow-up kind of like conversation. I can tell you, you and Leah, people really want to meet with you. You've mentored so many people, young students, on these webinars. And if we can get together to kind of like sit down and talk story, because they would be your go for it. In other words, I mean, there would be influence to kind of carry on the studies and the articulation, the research you guys are doing. I mean, that's the most important thing, you know, how we start looking at the next generation. I mean, you have inspired so many of us, and I appreciate that. Anyway, I also want to say,
oh, go ahead, sorry.
I also want to say your presentation has been so non-biased. I mean, you know, the facts and the problems, the Hawaiian and the other perspectives, the research, it is something that I've never heard. You mean, you just really have been able to professionally filter it out so that we have no other choice but say, hey, we got it. Mahalo, guys.
Wow.
Thanks, Emmet. I want to quickly go into just a couple questions that came up through the Facebook viewers. One was, how close to a possibility that genomic data will become an acceptable model for securing resource-like based financial exchange? I'm not clear what that question means.
Yeah.
Okay, well, let me go to the next one. My life insurance requires DNS to verify that you're a non-smoker, but they said they could not dispose of the sample after 2 years.
Yeah, that's an interesting one. You know, when we think about, you know, genetic discrimination, right? We know that, you know, there's a law, it's federal here in the United States that's called GINA. And what it is it basically, you know, protects you from any type of predisposing genetic diagnoses when it comes to two things. Number one, it would be to secure health insurance. And number two would be to secure employment, right? So, you know, you have yada yada disease, you cannot lose your job because of that. Yeah, Gina, exactly. Thank you, Keolu, for typing that in. So for things like life insurance or supplemental or disability or all of those things, those are actually not included under this, you know, protection law. And so if, you know, your life insurance did want to, you know, get your DNA, you know, smoking is something that's there's no specific gene for that. And it's certainly multifactorial, but, you know, it is possible that they could have been surveying for other things that are not under the protection of GINA.
That would not be cool.
No.
But that's, yeah, that's not cool, but certainly a possibility, unfortunately.
Sure. You know, I want to be respectful of people's time, and we are at the end of the time, but I wanted to ask the two of you, first, thank you so much. Thank you, thank you, thank you. And I want people to know that these two have volunteered their time, they've lent their expertise, they've certainly had to catch them at an eye up on a lot of things. So we really appreciate that. But because of the nature of the questions that we had in the evaluations, my two questions to you both are next steps for you both, and then specifically recommendations for the audience to keep the momentum going and to support the equitable paths forward. So you presented a lot of that, Keolu, but people really are saying, what can I do? You know, so to both of you, next steps and how do we keep this momentum going?
And you want to go first?
No, it doesn't matter. However.
Oh, okay. I mean, just the things that I immediately thought of is like, next steps is, if you don't think that a lot of these initiatives are good ideas, then definitely do your research, make your citations yourself, figure it out, see if you believe what they're putting down, and definitely vote, which looks to be very effective the last time, whether or not you believe that kind of stuff. But definitely with respect to like local policy is really important, and bringing this to the attention of our community and educating everyone about what the potential use and misuse of genetic information is, whether it's something you learned from our conversations or you want to follow up with us. And then I think that, as far as what we're going to do next, yeah, we want to build infrastructures, whether it's like a data science Indigenous Data Science Policy Institute that directly benefits the Kanaka community, or a biobank that directly benefits the Hawaiian community, or an Indigenous Futures Laboratory that benefits our community as a place for us to recombine ideas and think about our future as Kanaka futurists, just like Kamehameha, just like Kalakaua, just like Queen Liliuokalani, and the all-star incredible people that have motivated all of us. Like, let's move those agendas forward, so we're here for that. And I just want to say too, because I don't know if we're going to do a fourth one, but it's just been such a lovely but it's just been such a lovely opportunity to get to work with like our mentors, and for both of you guys, mahalo nui, because you guys are legends in the game. So we appreciate you too.
Yeah, absolutely. I couldn't agree with Keolu more. I think, certainly now that we've had this opportunity to kind of bring some of this to the community, I think going back, talking to different, groups, of course, the kupuna input is really important, bringing all of that from the research side to what's going on in different Native Hawaiian organizations, different healthcare systems, kind of building a think tank to then bring these things back to our community is really, really important. I also love that idea of the book club, Singh. I think, there's certainly a lot of work to be done. I think going back to the original question that JoAnn had brought up, earlier at the top of this talk, there was a question about whether or not it would be realistic to abstain from, genomics research. I think, like Keolu said, there are paths forward, and currently we are being left behind. I think mitigating the bad impacts is certainly what we're kind of geared up to do. And I think for me, my duty as a clinician is to kind of make sense of the things that we're thinking about, and again, move forward for equity for all of our patients on a system-wide level. And so I think those are all things that we're trying to work on together, and we're going to take it one step at a time, but having the input from the greater community is so important, and it's the only way that we can really move forward. So I appreciate everybody's attention, and of course all the comments in the chat. Really, really great questions, and I've learned so much from you, Keolu, and of course with Dr. Aluli and JoAnn at the helm. This has been such an awesome experience. So thank you.
Emmet?
So I'm looking at the next steps also. Leah and Keolu, we got to get you here.
Yeah.
Because we got to get you to talk with the communities. I mean, and to kind of like do the research as they would want to participate in the kind of research would make a difference. I mean, and that's JoAnn's expertise, yeah. and there's so many young folks that are so inspired, and to be able to kind of like meet up with them and continue to work. And, they're just kind of like, I can't believe how inspired they are, you know, like as we are. And if we can look at COVID-19 and how it impacts upon Native Hawaiian Pacific Islanders, I mean, what is it, you know, genomes? I mean, I think that'd be the first time anything like this has ever been done. Anyway, you guys are great. I'm so thankful to be young or old enough to learn from you guys. Mahalo.
Thank you. I want to again thank our sponsors, Papa Ola Lōkahi, Ahakane. Office of Hawaiian Affairs has been supportive as well. And I think we thank you for just your expertise in letting us move this conversation forward. I think I mentioned before that when we addressed this 20 years ago, we didn't even know what questions to ask. The issues were similar to issues around every other issue for Native Hawaiians. So that we knew, but this was an area, this scientific area was new for us. But at least people had the smarts and the gumption to just say, stop, wait, we need to learn more. And we had just great minds coming together over the Pauakalani Declaration to talk about the building blocks that should grow this. I'm so excited that you folks are moving forward and you have good models. I think we've made some strides, but not enough. I implore everybody who's on the webinar today to just be alert. We cannot be complacent. These studies are coming in. They're all for the good of mankind. We've heard that rhetoric before. We have to ensure that our participation makes sure that the benefits extend to the people who we care about and the world. So thank you to Leah and Keolu. It's been such a joy to work with you guys. I don't think it's over. One of the questions on the evaluation is, what else do people want to know? So hey, maybe that's bait in the water for future sessions. But thank you. And thank you to all the viewers and the lovely comments. I do share everything that you report on the evaluations with our speakers. It's largely been positive, almost all of it, but it's been provocative as well with the questions that have come up. And that's very guiding for these two young folks. Us old folks got to sign off now. So I want to thank Kim and the staff at Papa Ola Lōkahi for running the behind the scenes.
Mahalo, Kim.
Yes, Kim. And POL team, you guys are fantastic.
Yeah.
Yeah, the whole team of students and, you know, thank you all. So Ahahui ho, yeah, I don't think it's over.
Right.
Everybody have a safe trip home.
See you at least 3 or 6 months.
Yes,
JoAnn and I worked on it.
we're already scheming.
As soon as it's safe.
Yeah.
Yes,
Right.
we're already scheming. Yeah, and we'll use technology in the meantime.
Roger.
So is Kim still on?
I don't know.